Alrighty, everyone. Thank you very much for coming today. I'm Albert Radloff, the PBC Center Coordinator and a graduate student here in the Anderson Lab. Thank you very much for attending. A few quick housekeeping items. We are sending out updated email lists, surveys. Sorry for the annoying PBC emails, but please do fill those out so we can make sure you get relevant information to you. And we're also going to send out a survey on speakers you'd be interested in having. These seminars are really important for the students and the faculty. We host our guests and sometimes their spouses as well, other distinguished researchers in their respective fields, for a few days to meet with faculty and with the students, so we can all have a kind of a collaborative learning environment. So any folks you are interested in that are related to the plant breeding field and help fulfill the Plant Breeding Center's mission, please do put them into the form once you get them. Today, we have two great scientists with us, and we'll be having a seminar from Dr. Trevor Rife here, who started at University of Wyoming, then Kansas State for graduate school, some postdocs, and then started what we're all here to learn about, field, I just completely spaced, field book, I apologize, and breed base adjacent.
Yeah, not really. But
We'll learn from Trevor today. So he also has some free time this afternoon if anyone would like to meet. We also have his partner, Dr. Jenna Hirschberger, who's a vegetable breeder at Clemson as well. So feel free to reach out and we can schedule something for this afternoon, but I'll leave it to you.
okay, thank you very much for the invitation. This is a pretty big honor, and having been on the other side of the, watching seminar speakers as a student, it's kind of trippy to be on this side of the desk, but I'm really excited to be here and talk about some of the work that I've been putting together for the last many years. We had a great conversation with the students yesterday. I think we talked for like 2 hours, which was pretty crazy. But one of the things that did come up was just geography, and I figured that starting out, it would be good if my slides would work. Good to talk about where I actually am. And so this is a map of South Carolina, Georgia to the south, North Carolina to the north. Clemson is up here in this side of the state, and then has a network of research stations that have about a dozen faculty each, one of which is Florence, South Carolina, very different from Florence, Italy, but on the I-95 corridor. So we're in the Midlands of South Carolina. We have this really strange building that I don't think would be allowed to be built today that spans a pond. It's really cool. Our offices are just looking right over into the water, and so we get to see all of this fun flora and fauna, including otters that pop up occasionally, lots of turtles, lots of fish. Occasionally we get a few other guests, and so we'll zoom in on this guy swimming like right under the building. And so we had a resident alligator over the summer, which was different coming from Wyoming and Kansas to having alligators right out the window. But that aside, my program mostly focuses on looking at the breeding cycle, figuring out how we can optimize this breeding cycle, mostly on the data collection side. And no offense to the breeders in the room, but breeding is a simple process. Logistically, it's not, but the actual process is fairly straightforward of selecting material, intermating those materials, and then evaluating that material over a long amount of time and space. The thing that ties all of these steps together is the massive amount of data that's collected at each one of these stages. The data collection hasn't really changed a lot. Going back to Borlaug at Summit, when I started my PhD at K-State, really didn't look that different. But the breeding process had really changed. And so we had these much larger populations, many more phenotypes that we were needing to collect to make those genetic gains across all of the material. And so one of the big steps that I took during my PhD work, during my graduate work, and that I've continued as an assistant professor is working on tools to digitize that data collection. We have a bunch of these different tools. I'm going to talk about hopefully all of these if I don't run out of time, but spend the majority of my time talking about Fieldbook, which is probably the most successful and most well-known of all of these tools. And I will say, you're going to see a lot of pictures that look like this, but this is not what it started out looking like. If we go back in time a little bit, you can see that it gets worse and worse as we go toward the beginning. And I think that this is really just indicative of how important it is to get something done and iterate on it as quickly. 12 years isn't that quick, but as quickly as possible. You really can't make improvements until you have a starting space. And so while this kind of was a little ugly and kind of had issues, lots of them, we've really made a ton of improvements. I'm going to talk about some of those today. So Fieldbook itself is a pretty simple mobile application, runs on Android, totally open source. So the basic operation of this is that we can move between different entries. And so these black arrows move between our different plot numbers here, going forward or going backward. We have different contextual information for each plot up at the top that we can import. And so this is really adaptable to pretty much any breeding program. We haven't really had one that has not been fixable into this framework. And then underneath that, we have our different traits navigation. And this is these optimized layouts for plant breeding, data collection. So in this case, we have something like a counter trait, a numeric trait, a percent trait, and a categorical trait. And these can be mixed and matched, customized to really whatever data types you need to collect. And one of the things we've been doing recently is starting to tap into some of the sensors in the device itself. And so we released this Monday, so actively working on all of this stuff, which is really exciting. But we have this new angle trait, and so really valuable for people like Soon Lee, who's not here, but capturing like branch angle or corn leaf angle. A lot of this is done potentially manually or via image analysis downstream. A grad student at Iowa State actually captured all of his images in Fieldbook and then had students, undergrads, process all of those images, drawing little angles. But in this case, we could have just captured the angle automatically. Not helpful for him, but helpful in the future. One of the other sensors that we've really been tapping into is the camera. Capturing image data is notoriously great, but also awful. Thinking about historically capturing that data, a lot of people would use point and shoot cameras. You'd have to track the order of the images or take a label before you captured the image. With Fieldbook, so taking one of these images, the image is named for the plot that you're on. It's organized, it's labeled. You can collect images from different sources. So we can plug in a USB camera, we can hook up a GoPro. We also have some compatibility with certain Canon DSLRs. And I think what's really cool here is that every image that we capture in the EXIF tags, the metadata for everything related to that image is stored in that. So even if the file is renamed or processed into something else, all of that information is still saved within the image itself. And this is some of the initial things that we've been working on in the most recent version that came out in January, version 6. One of the other big things was just this ability to manage lots of fields. And so this is some sample data that this is real data that I got from the wheat breeder at Virginia Tech. And so he had 40 fields loaded, which were a little difficult to manage, but having that real data makes things a lot easier to see how people are actually using the app and make improvements. And so one of the things that was obvious with 40 fields is that we need a way to really sort through these and to process these in bulk. So we added this ability to sort by the name, by the format that was imported or when it was imported, when you last edited it, when you last exported it, all these different parameters. We also added this ability to bulk manipulate these files, so we can select multiple ones, export them as this giant CSV or zip file with multiple CSVs in it. And this was one of his biggest complaints that he talked about at PAG, like repeatedly. And so I do listen to people who actually use the app and tell me about it, tell me about their issues. And so we can also search for specific fields. And in this case, we have this new field here, and we select that. This is also a new addition in version 6, having this sort of independent field page. So we can expand some of these different cards and see additional information. We can rename the field if we need to. That name is generated based on the incoming file, but sometimes you do want to change that. Or we can sort the field by, or the plots within that field by different parameters. We also have this data card. Oh, whoops, there we go. We also have this data card at the bottom. And so expanding that, we can see the trait that we're actually collecting in this field. In this case, emergence, the percent complete. We have only 8% of the phenotypes, which I like to give Nick Santoni a little bit of crap for. And then the distribution of those actual phenotypes. And so we have that really good visual check for the data that's coming in that we can then see and make decisions. One of the big things that we've focused on in the last few years is adding breeding API compatibility. And so without going into too many technical details, the whole informatics breeding community came together about a decade ago and basically defined every parameter of what breeding looks like. And so this common language that we settled on is called the breeding API. And what that fundamentally allows us to do is to take a client, whether that's a computer or a cell phone, and a database, and then automatically communicate back and forth between that. And so since we have that defined language, we can send and receive data matching that spec, and then just write our translation layer in our own tools. And of course, in this case, we're talking about Fieldbook as the client, but then on the database side, the cool part about having this API is that anyone that integrates this is then compatible with Fieldbook for collecting data digitally. And so some breed-based, a lot of breed-based users, BIMS, which is developed by Dory Main's group at Washington State, and then other tools like Delta Breed from the Breeding Insight Group for USDA, the Enterprise Breeding System with the CG network, IBP, which is from the Integrated Breeding Platform, or BMS from the IBP. And then, which is, I think is the coolest, is that this also allows private groups to do the same thing. And so BrewGen is a product from AB InBev, and they designed their own breeding database, but all of their breeders were already collecting their data with Fieldbook and didn't want to switch. And so they added RAPI compatibility, and so the data can move back and forth between their system and the app. And within the RAPI functionality, importing fields via RAPI and importing traits, we can do a lot of things. We can search, we can filter based on things like the program or the year, season, things like that. And so this is an example using Cassava Base, which has 7,000 fields from all of the cassava breeding programs in Africa and South America, and filtering that down by IITA in 2023 season. We still have 140, but we can do that really quickly with this type of approach, and then import multiple fields to then go collect that data, push it back to Cassava Base. Same thing on the traits. So this is a really strong way of maintaining ontologies, making sure that everybody is collecting the same data the same way. And so that when you go to put together all of that data, or when it's in the database, everything, all the columns match up. So we were talking about this during the lunch yesterday of working with historic data and getting those columns to line up. And if anyone's done that, I'm sure most people here have at some point, they don't line up very well. But this is a way of maintaining those standards for everybody using the tools. Of course, once data's been collected, then we can push that back to the database. And in this case, I just collected this random observation just to show it as an example. But really what we're doing is completing this digital data ecosystem and lifecycle. We really don't want to move data into Excel files. That is sort of like the enemy. I'm not as adamant against that as like Jim Holland, who I think is even more extreme against Excel. But anytime that we do move files into or move data into those files, it's an opportunity for mistakes to happen. It's really easy to click on the wrong cell or type in the wrong thing, copy the wrong column. But doing things in this type of approach eliminates that opportunity for mistakes. All right, so 20 minutes of background so we can talk about the cool stuff now. So we've been working on Fieldbook for, it's almost 15 years, it's more like 14, but and over the course of that time, we've designed this phenotyping app with these base traits, these base layouts that have been built in. We've kind of supplemented that with these custom traits, so things that are really customized to different workflows, different breeding programs' needs. But we're really not capturing a lot of the phenotypic data that we could capture. And so what we've been thinking about moving toward is sort of adding in modalities of data collection. So whether that's from different external devices, via image analysis, pushing off heavy analysis loads to things like remote servers, for running like machine learning models or anything like that. And I needed a last one. So analytics is pretty generic and kind of captures everything else. But all of these different data types are things that we aren't doing in Fieldbook that we can. So I'm going to talk a little bit about how we've been pursuing some of these different avenues. And that really starts with themes. And so more than just for branding, I'm not going after like the Coca-Cola or Mountain Dew money here, but we spent a lot of time updating Fieldbook to be able to theme it. And there was a really specific reason for that we wanted to get to this nice black and white, and this was sort of a linear progression. And the reason for this is that we identified these new devices that are the perfect device for plant breeding. I've been looking for this for like 10 years. I've ordered so many devices off of AliExpress that were not the perfect device. And these are called Books Palmas. They have a camera for barcode scanning, which is still decently good for taking pictures, but more importantly, they have an e-ink screen with a pretty high refresh rate. And so the battery life on these is insane. I just turned on the one that I carry around. It still has like 95% battery just sitting in my backpack since I honestly don't know the last time that I charged it. But I think really importantly is that this is how they look under direct sunlight. So we have a normal LCD phone on the right. We have this Books Palma on the left. So this is the perfect device for phenotyping. I wouldn't be saying it if it wasn't true, because I have a stack of them that it wasn't true for. And it's like about $300. And I'm not making anything from these, but it's great and it's available. And we've spent a lot of time optimizing Fieldbook specifically for this because it is so great. Of course, there are other external devices that we can work within too. And one of the big things that a lot of breeding programs have been doing over the last 15 years or so is relying on high precision geospatial RTK GNSS systems. So we get that centimeter level precision that we can use for activities like planting, which that's where most breeding programs kind of stop using those, but they still have that hardware that they invested in. And so one of the thoughts that we had a while ago was that we could just tie that into the phenotyping process. And so this is an older model. These newer models fit on these really nerdy looking backpacks that Keo and Heather are wearing here. In this project, I like my acronyms because I've had too many federal projects, so we call this halfback. But putting the actual location into the breeder's hands, and then using the location of the breeder with these GPS systems to then track where they are, connect that to where the plots are, to then make phenotyping easier, removing that navigating step that can actually really mess up where the data is being collected. And so as an example of this, this is a wheat field at the PD Rec. So Rick Boyles is our small grain breeder and is kind enough to let us play in his material, which is really fortunate. I'm just going to be focusing on this little red outline. I don't think anybody wants to see me like phenotype or walk through all of this, so I kind of cut it down a little bit. But the process for using this type of tool is to first collect the coordinates of each plot. You can also import those coordinates if you have them from like a planter, which, yeah, so it kind of making that full life cycle of the data as well. So here you can see me surveying each plot in that little block. And then this is a fairly, it's not long video, but we'll have time to kind of go through everything. I'm down there in the corner, right here. On our screen recording here, we can see the range in the column that I'm currently on. I've collected a few phenotypes in this field already, but I'm not going to be interacting with the screen. And so you can pick what you want to watch, but as I walk through the field, you can see that our column number is incrementing as I pass each individual plot, and I'm not touching the screen right now. This is all happening independent of everything else. So as I get to I think we're 4x15 is what I put together. So yeah, walking to the end of the range, getting to that 15th plot, and then just walking up to the second range. We just hop up to the second range, still on that 15th plot. And this is where I wish I could like fast forward sometimes because kind of like make it look like Benny Hill or something. But yeah, so this is the output of this. I'm still kind of amazed that it works this well, to be entirely honest. I feel like things break more often than they work in tech demos, and we got this to work really easily. And a lot of other people have as well. And so I'll get back to the first plot. We have two border plots in this case, collect a phenotype, and that was the first time that I actually touched the screen during the whole video. So really cool stuff. Maybe not as relevant in the wheat world, but in something in a perennial like apples or grapes or nut trees or peaches in this case, it's really important to be able to know where your trees are, especially when certain trees die and when the tags on those trees are exposed to the elements over the course of many seasons and often disappear. And so having this type of system available means that the peach breeder at Clemson, who last fall went through and phenotyped or surveyed all of her trees in all of her fields, is really important. And so she can just walk up to any tree now and collect data, which is really cool. So moving around the circle a little bit, image analysis is probably one of the areas that I'm most excited by to integrate into Fieldbook. And so I'm not going to talk about it a lot today, though. We're going to go back to external devices. But I wanted to actually see if image analysis, how hard that would be to integrate into these types of tools. And so I had my developer, I basically told them just see how difficult it's going to be to integrate something like Deep Lab v3 or MobileNet directly into the app. And it was pretty straightforward. And so we have a segmentation on the actual image preview happening here. And so we can successfully phenotype cat in Fieldbook, which is pretty cool. But I think so yeah, the analysis side, I think is going to be pretty easy. And so then it kind of swings the pendulum back over to how are we going to collect all these images? Because we need this to happen at scale. And all breeding happens at scale. And so things that work for genetics research projects don't necessarily scale to the breeding level that we need them to be at. So fortunately, I had the opportunity last September to run into Corey Hirsch at a meeting in Fargo, who talked about some of his work with selfie sticks. And that like sent me down this rabbit hole of this is the way. This is a great way of capturing images. You can control them, but we don't want to have to interact with that screen. And we do have the GeoNav, so we have that as a potential opportunity. But then I went down this other rabbit hole of finding these Bluetooth remotes that also exist, mostly for cycling and things like that you can strap onto the handlebars. And so it's really easy to modify Fieldbook to just work with these. And so we can navigate plots with our volume buttons. We can go to the next and previous traits with our arrow buttons. And we can capture pictures with this play pause or the call end button. And so this is really a cool way of capturing images. And so just to demonstrate that, we're going to go back to the same field. We're going to go this way instead of those 4x15. And then this is going to explain what's happening because I've been told this happens too quickly, which I take as a compliment, honestly. But we have our preview right here of the camera or what we're actually seeing in the viewfinder. As images are captured, they're going to go off to the left. And then we have our row and our column information right here. And so as I progress through plots, I think our column is going to change. I think the column changes in this example. And so I'll play that. And yeah, the screen recordings are a little janky, but it looks better in real person. And so as I capture those images, they're saved off to the side, taking 2 images per plot. And then as I get to the next plots, I hit the next button and go to this. And so again, not interacting with the phone at all, but capturing images at the pace of walking. So we can scale this with undergrads, right? That's the answer. We scale it with undergrads. We can collect images on everything pretty quickly. And while this is maybe not the best example because these overhead images could be processed with a drone, for things like beans or any other species where it really matters the side of the plants or the under the canopy, that data can't be collected with UAVs. And also the whole fact that the UAV data still needs to be processed. So we're really moving toward this idea of being able to capture those images, process those images, and have the results all in one place without having to go through any other steps. These are obviously not the pictures from last week when I collected this. I did this also in January. And so we can easily see some pretty good emergence or ground cover differences in this case between 2 different plots. And so my rule of thumb when people ask me about image analysis is if you can see it by eye, then you can probably figure out a way to do the analysis. So yeah, we didn't do it, but obviously this would be trivial to exercise left for the reader, I think. But a lot of times we just want to capture an image on an individual row for things like disease nurseries or hedgerows. And so one of the things that we also added based on that conversation in September was this ability to crop the incoming images. And so in this case, it's maybe not the best or most visible, but you can see that this side of the image preview is a little darker. And that's because I cropped all of the incoming images for this specific trait. And so I'm only capturing an image of each individual row and then going to the next row. And we can see that in this case it says column, but examples. Yeah, very cool. All right, so this is where we're at. And I guess the nagging question that I've had for the last 15 years is, are we making progress? Is today better than 1970? And so we wanted to actually find out if the steps that we're taking to make breeding, that I think we're taking to make breeding better, are actually having an impact. And so also in September was busy for me, I guess. We did this experiment in the sorghum association panel. So very diverse panel of sorghum that we had planted at the station. In this case, each row is a different genotype. And what we really wanted to do within a small subset of that panel is to figure out if Fieldbook was faster than paper recording for phenotypes. And we also threw in Google Sheets just to see if can have a comparison on the digital side, talk about some of the maybe limitations of that. But within each one of those rows, we tagged one plant, and then we just collected 3 phenotypes on that plant. And this was a really weird experiment to set up because it's kind of hard as like a plant person to like not care about the phenotypes and be working with people really, but we really care about the moving between and the writing of the data down. And so we wanted to make it as easy for the people and not collect, the number of tillers and 20 plants that really like to tiller. So we really limited the data, the actual data that was collected. And so the 3 traits that we collected were the number of tillers on one plant, the flag leaf width, and then the length of the panicle. And between all of those different methods, I think good sign was the phenotypes generally didn't vary a lot. There wasn't like a method by phenotype interaction, which would have been bad. But ultimately, the crux of the issue is that this is the raw data. We're still processing these results. Is that the digital methods were faster than the paper methods. And this is, like I said, raw data factoring in person and the order in which things were collected. These are statistically significantly different. The obvious reason for this is that when you write things on paper, you have to transcribe it later. And so there's an extra 15 to 20% added on to the end of that collection process. And you can see the differences between total times for phenotyping by a lot of these different people. Person number 2 is not me. It's a sorghum grad student who is awesome and knows exactly what she's doing when she walks into the field. So one of this says Excel, but it was Google Sheets. One of the outcomes of this was that, sadly, Google Sheets and Fieldbook were not different. It didn't really matter how you collected the data. As long as you did it digitally, it was going to be faster than paper. So a little bit of coping with that, but one of the things with Fieldbook is that it is optimized for those larger populations. You also get a lot of that metadata with each observation that you're capturing, which is really interesting to go through and look at. You can do some fun analyses with these. And so I hadn't done this until this talk, but this is data I collected during my PhD. This is just height data from one of the experiments that I did using one of these bar-coded height sticks. This is the picture of my advisor that I always put in every talk when I talk about these bar-coded height sticks. But you can see that with this metadata, these time between each measurement that I captured, there's these weird little outliers up there. And when you go and look at the actual data, that each one of those is at the beginning of a different row. And so what had happened was that I got off. As I was counting each individual plot and collecting a phenotype, I would lose count or I would miscount, and I have to go back to the beginning of the row and start over. And it's very apparent here, but it only added like an extra 50 seconds. I've talked with other people where they see like 10-minute differences between phenotypes. And it's at that point, you know that somebody's maybe taking a phone call or slacking off a little bit more. But it's really cool to be able to do these types of meta-analyses with this data. So I alluded to a few of the other apps that we've worked on over the last few years. And so I'm just going to kind of go through those fairly quickly to talk about some of the other aspects that we've thought about within breeding and within genetics. So a lot of the data, a lot of the samples in this field are organized into grids. And we do that so that we can be higher throughput, or so that we can more easily plant things like hedgerows. But ultimately, keeping track of the samples is kind of difficult. You either pre-plan the sampling, create pre-planned sampling maps that you have to then match samples to, which is a opportunity for error, or things like that. And so we developed this app called Coordinate that's absurdly customizable to a fault. And you can create different templates, and from those templates, you can create these grids that kind of match exactly what you need in your own breeding program. And so in this case, I have a genotyping plate pulled up here. So we have our A through H and our 1 through 12 with just the 9 through 12 being cut off the screen. And the idea here is that as we put a sample into our well, we scan the barcode for that sample and we know exactly where it's going. Export this to a CSV. That CSV is our key file for downstream analysis. It all links together, which is really nice. Say one of the core tenets of plant breeding is controlled crossing. I don't think that that's a contentious thing to say, but a lot of the crossing activity ends up looking like this. And so this is from the cassava breeding program at IITA. Writing on small tags, a lot of information, and then transcribing that to larger envelopes, along with metadata like the number of seeds that were harvested. I think this is the number of flowers that were pollinated. And so all of that information is, and generally the people who are doing one step of that writing and are not doing the transcribing or the harvesting. And so there's all this opportunity for mistakes throughout this entire process. We worked with a cassava-based group and the Next Gen Cassava group to move the IITA cassava breeding program to something looking more like this, where each plant is barcoded, each pollen bag, in this case, it has its own barcode. And with this, we can start thinking about how we can track all of this information digitally. And so we developed this app called Intercross, that pretty straightforward app. You have a male box, you have a female box, you have a cross ID box. And this is the default configuration. This can be customized to whatever you want it to be, pretty much. If you want to capture the male first, you can do that. If you want to have a blank male for an open cross, you can do that. If you want to supply your own cross IDs, you can do that. And so I'm sure we haven't met everybody's needs, but we've done a lot to customize this to a lot of people's crossing needs. As you make those crosses, those individual crosses show up in this bottom list. We have our two parents, the dates, the cross ID, the person that crossed it. What's really cool with this is that we want to maintain that whole digital data chain throughout this entire process. And so we can connect this to this portable Bluetooth printer. It's like $500 that we can print off those cross barcodes as we're making them to have that barcode on the actual cross, have the ID, the parent information, and everything else. We can also import wish lists, import parent lists. So we know what we want to make our crosses. We know which crosses we want to make. In this case, I'm making two crosses between two things that I shouldn't be making here, Kharkov and TAM 107. And so I think Jim's probably laughing, but that's example files. But we know how many crosses of this we made. We know who made those. We know when they were made. We know what our wish list status is for this. And so my wife works on lima beans and recently looked at the crosses that had been made for the year. And there were a lot more crosses for these two commercial lines than there were for any of the experimental lines. And so knowing when to stop is really part of the really big part of plant breeding and cross management and making crosses. And then finally, I'm going to talk about Prospector very briefly. And so this was an app that I worked on during my postdoc. Connects to these portable spectrometers, relatively low-cost portable spectrometers. Then we can collect the spectral data in this really organized fashion. So only reason I'm not talking about this more is because we recently had a project funded to integrate these and other tools directly into Fieldbook, really that one phenotyping app to rule them all sort of model. And so bringing that back to our external devices, looking at things like spectrometers, green seekers, or colorimeters, that we're really integrating that data directly into Fieldbook so that our sample management, our phenotype management is all in one place. And so I wanted my developer to I kind of let him loose on this about a week ago, I guess a week and a half ago. The low-cost colorimeter, which is about $100, arrived at his house on Friday, and so we have some really preliminary stuff that he sent on Monday. But we have made this. Well, this is not a mock-up. This is a screenshot of what we have working right now, but it's not released yet. This is, I think, the only thing that's not released, and the cat model. But so this is coming soon. And then just yesterday, he sent this screenshot of the Inaspectra integrated as well. And so really designing this entire, making it, yeah, the cool part about all of this is that as soon as this is integrated into Fieldbook, outside of having to get the devices, as soon as any of these tools or technologies or approaches are integrated into Fieldbook, they're available and they're accessible to every single person using the app. And so that's about 7,000 people spread across the entire world. Lots of users in India and Brazil and the US. And then the other countries that you'll see kind of highlighted are all of the CGIAR countries, Peru and Nigeria and Mexico, but really a good distribution of people all over the place that have really made this possible and given me excitement to work on all of this stuff over the last few years. And with that, these people are the ones that do all the work. I just come and talk about all of the cool stuff that they do. And specifically, so Cheney, Prasad, and Brian are the 3 developers in my group. And then Walker, of course, my student took all of the drone data or drone images for the videos and everything else. Many other people have contributed code and ideas and just discussions to making all of this better and to making it available for everyone. And so I think with that, I can take some questions, either from here or online, but thank you.
For a nice talk. I've been following Fieldbook for many years. So one thing when I was hearing your talk, it was great to see you said, see the progress and all that stuff. And you said, and then we're going to have the scale. And I was surprised to see selfie sticks in undergrad, the solution at the scale. I'm wondering, have you given some thought about affixing your software effectively to small little bots that basically go up in the aisles? I'm not going to say that every phenotype is collectible that way, but there are some phenotypes I think that UAV would be good for, particularly height and things of that sort, that little bots would be very useful for, and then some things would have to be at this stage with humans. But have you given this thought of like that?
No.
Okay, fair enough. Fair enough.
No, I think that there are some groups. I think there is a BMGF project that is kind of looking toward that direction. Maybe not of bots, but of kind of wheelbarrow-like carts that might use external cameras but connected into Fieldbook to process everything. We're not there yet. There's a lot of other things. I think we have like 130 open issues that on GitHub that are more ideas than they are problems. There's a few problems, but more ideas, and a lot of those could be expanded. So there's a lot of other stuff to work on, which is one of the reasons that we haven't done a lot of things, including like an iOS port or things like that. It's just there's enough other stuff that we're still getting done that when we do accomplish that, we can start looking at some of those fun things. Which I think that's one of the big reasons, like the sensor stuff. We move really quick on that. Like that's like in the last week, all the sensor integration, the colorimeter and the spectrometer. But it took all of the like planning and everything before that to actually be able to move that quickly.
Yeah, that's great.
Yes.
Would it be possible, do you think, like record voice, like saying out loud the number, like walking down the field?
Yeah. So I guess for online, the question was recording voice. We do have a trait type that does record audio, but it doesn't transcribe it. So it's kind of like I don't want to say useless, but less useful is the way. Yeah, I'm afraid I have removed things from the app before and received an angry email that I thought, I was like, nobody uses this. Like, this is, we don't need this. We're just going to take this out. And then somebody emails me like, I am in the field and I cannot do this right now. And so I just can't remove things. We have a lot of interesting ideas for voice integration, but also for like some of the newer technologies like heads-up displays and things like that. I'm in the process of writing some proposals, so I don't have funding for that right now, but I have ideas. Yeah.
What's your read on databases?
This is being recorded, so I'm going to choose my words very carefully. Databases are really difficult to set up, period. And a lot of that is that you can really easily mess stuff up if you don't do things in how, like, there are a lot of checks that have been put in place in software projects to ensure that it's harder to mess things up, is probably the best way to say it. So I don't know, I'm not answering your question, but
Like, do you have any ideas for like that's like the biggest issue that I feel like is in a breeding program. You just end up with so much data.
Yeah.
That's really disorganized, and like there's a number of databases that are
so I guess
all have their quirks. And
yeah.
As you said, like as soon as you import the data and then you try to get it, like then you like export it, something's changed and there's another step you have to do downstream to fix it.
I guess my probably most political answer to that question is that any database is better than no database, even though every database kind of sucks. And so it's really easy to move data between data. It's really easy to move organized data. That's like the end of the sentence. And but it has to get organized somehow and somewhere. And so yeah, terrible advice, but probably suck it up and choose a database and then keep looking out for one that's better, but just like go down the road. A lot of the quirks of the databases that are popular, like everyone's struggling with the same quirks and there are some solutions to that. I think that like with Fieldbook, there are things that, so I'm supposed to, yeah, I have Fieldbook examples because that's what I work on, but there are like things that people have found ways to get around some of the limitations, just they're creative. A bunch of people working or having their PhDs that, yeah, they're clever and they can figure stuff out. And database management and database design are the same thing. And so they'll have their quirks, there are ways to get around them, but ultimately just pick one and go with it and hope that something better gets developed at some point.
You don't want to work on it?
Nobody wants to pay me to work on that.
Yeah.
I would love to work, that's like one of the dreams is like designing a whole system. Having talked to some of the people that have designed those and some of the like, I think Breeding Insight, the USDA database system, they have like 70 different breeding schemes that they have to support across, I don't know how many dozens of species they're working on now, but that includes animals and like honeybees and salmon and trout. So yeah, I would love to work on it, but also I don't think that, yeah, we'll see, maybe someday.
Nice talk. You mentioned support model, so I think all the ideas you have there are really great. And if you look at things that are all different technologies that you're talking about, of course, technologies fail and we've got problems. So what's your vision for like the tech support model? Very constantly see yourself using tech support calls all the time.
Fortunately, less than I used to as like a student. I still get emails with problems. And that's probably not where I should be spending my time, but it's like, it's what I like to do. And so I have people that I could hand off a lot of those issues to. It's probably not sustainable in like the super long term, but I'm passionate about this and so I care. And so anything that goes wrong, I kind of take personally and want it to be fixed.
Long term, I mean, like if everybody adopts this new technology.
I'm hiding this. Been really fortunate to have community support and funding support from from a lot of different agencies and different groups. I've talked with a few private companies about possibly them supporting development as well, but maintaining the open, kind of the free and open source system that we have. Those conversations are ongoing, so I don't want to say too much, but I don't know. Like ultimately, it's all open source, and anybody could take it and pick it up and run with it, even if I'm out of the picture, or if the funding for me is out of the picture. Which I think is probably one of the most important things for a software project, is that if the source code is not available, then you are really beholden to whoever is signing those releases, because as soon as they disappear, then whatever bug pops up or incompatibility with the new system pops up, you're kind of screwed.
I think with Fieldbook, sometimes if you just don't know how to do something, like it's totally possible you just can't figure it out and you need to talk to somebody. And so having someone you can talk to, be like, how do I actually do this? But if you had a lot of people using it, even if you had like an AI assistant that would like walk people through.
We've talked about that. Again, other low-hanging fruit that we want to do first, but totally having like support built into the app would be a valuable way of doing things, or potential way of doing things.
Recently updated your document.
Oh yeah, and we have really good, robust documentation as well. So you can get to that from the phenoapps.org page that I linked earlier, or that I had a picture of earlier. It's pretty thorough. I think that it covers most things, but also like, yeah, maybe to my detriment, I'm okay being an email away. And so if there is like a challenge or something that pops up, then that's often a 30-second email for me to solve like a 3-hour problem for you. So I think that's a fine trade.
In one of the slides where we mentioned that we could take image data directly into and I saw the icon of genomic like on that how that's and then when you walk through the fields, and then you might have to like again it captures a single image of the whole plot at the moment, and when you have a sheet that you uploaded on the feed book, collect the data, and then you have multiple images of the same plot. How does that handle?
Yeah, a lot of questions there. So the Canopio app is a separate thing. It's just the iris capture button is what you saw. And I think that just does like ground cover, calculates ground cover, but it's mostly targeted toward producers, like more than readers and scientists. So that's one aspect. That being said, having the analysis tools built in is definitely the dream. So moving toward that model. The image capture, the images are separate files from the actual data that's being collected. So the images are saved in a directory, but they're also named with the plot ID, timestamp, and then they have all of that metadata embedded into them. And so then within the file that you export from Fieldbook, you have a list of the images that were associated with that specific plot.
We need these coordinates. Like how you show these coordinates of which if we capture that image, does that help?
No, that's only specifically for the GeoNav system. And so if you want to be able to navigate automatically between different plots, you need to know where those plots are. And so, but only for that. So that's just one of the features that is available.
Real quick, a few online questions and a lot of thanks and appreciation for your seminar. A simple one here, is it free to use?
Yeah, totally free, open source.
Okay, and best thing to Google or website to go to get this updated documentation?
phenoapps.org.
Alrighty. And then any thoughts on a desktop-based version for more interaction and analysis in the lab outside of an app or tablet?
I'm curious who asked that. Probably no.
Okay.
I think that we have like this nice niche cut out of sort of this life cycle. I think like having the overarching summary statistics that are built into the app is valuable, but doing any of the actual analysis is definitely outside of scope. And that's kind of like not going to probably do that anytime soon. I think that's what belongs in a database systems and things like that.
Excellent segue. The next question was, does the data, and we talked on it yesterday, so when you collect data in the field, you can immediately export to the cloud in that moment.
So
And does it go to a database at that moment as well, if it's already integrated?
depends. Yeah. Short answer is depends. Long answer is if you have a connected database and you have internet connectivity, then you can send that to a database. If you don't have that part set up, when you export, it exports to a flat file on your device. So we were designed this entire system with offline first in mind, and but from that you can upload that file to the cloud pretty quickly.
Any Les?
A couple of things. That favorite Android device that you were talking about, is that a phone? I couldn't hear what the name of it was.
Yeah, so it is this guy. I put it in my back pocket just for your question. If I can get to a nice spot where I can.
What's the actual name of it?
The Boox Palma, BOOX Palma. And it's also on the phenoapps.org website. I have a hardware page on there that I update once a year-ish of like suggested devices. Just because I get this question a
It's
lot.
a regular phone.
It is a SIM-less tablet or like a SIM-less phone, I guess. It doesn't have a SIM. So network connectivity is not there, which is a plus in some cases. They don't walk off as much as phones do. But it's phone sized, but doesn't have
More like a
a,
tablet.
yeah.
And the other question, could you capture the initial block coordinates remotely?
Yes. Yeah. And so from what I showed, we were surveying within the app itself, but you can also import those coordinates when you're importing your actual field. Yeah. It does, yeah, it has a little tiny camera on the back. The quality is acceptable. It's like 16 megapixels, I think, if I remember correctly. It's not the best sensor. It's definitely designed for like document scanning. It's the perfect device, but like, yeah, the camera kind of sucks, but it's. But yeah, that's one of the big reasons we did the external cameras is because those e-ink devices were like the ideal devices, but none of them had cameras. And then they released this.
Good question about the books.
Yeah.
It doesn't have, so it does not have connectivity, cellular connectivity.
Yes.
So then how, can you use it with the GNNs?
Yeah, well, mostly.
Cause I do the triangulation with like a MN cores, like, or MDOT base through the cellular connection on my phone.
So
We don't have a base in our field.
long answer is it depends on what system you're using. If you have the base in the field and the rover in the field, then generally it's rover to phone Bluetooth.
Yeah.
If you have, we have done it with the ones that do require cellular connection, but we generally just open up a hotspot on that one and then connect from this to the hotspot over Wi-Fi and then have access to the coordinate
Okay, so
correction.
so from your phone. Okay.
Yeah, there's a phone coming out with a e-ink screen that does have like everything, but I don't think it's out yet and I haven't tested it.
I was curious, kind of off that note, and with some of the sensor talk, if I had a new sensor maybe I made in my lab, would I be able to set up Fieldbook just with a generic data collection template so I could get all that metadata and easily just through ID number connect it to my sensor that I'm running separately?
Not currently, but that would be something that we're trying to move toward that model, but getting generic connectivity things in place is there are a few of them is sort of the short answer. That was a problem with the camera stuff, was that none of the cameras use the same like connectivity standards at all.
Alrighty, folks, do we have any other, oh, Jim, is that a hand? No. Any other questions? They do have some free time for the next hour. But please do give a round of applause and thanks.